Have you ever wondered about how to change hormones or brain size? Do you think sea slugs are cool? Well, today I'll be talking about all of those things, so listen on if you'd like to learn more. Today I'll be presenting about how the number of vasopressin immunoreactive cells correlates with anatomical size and condition in the nudibranch Berghia Stephanieae. My name is Meagan Olson, and this work is done in collaboration with my mentors, Dr. Cheyenne Tate and Dr. Paul Katz at the University of Massachusetts Amherst. This is the sea slug I'll be discussing. Its name is Berghia Stephanieae. Here is a photo of it with some labeled body parts. It is classified as a mollusk, gastropod, and a nudibranch. It is relatively easy to raise in a lab, and it matures quickly. Berghia has a hormone called conapressin, which plays a role in their reproduction and the reproduction of many other species. On the left is a photo of two mating Berghia. Conapressin is homologous to vasopressin and oxytocin. On the right is a comparison of amino acid sequences of these related hormones. In this experiment, we've actually used vasopressin immunohistochemistry as a proxy for conapressin expression. These are pictures of Berghia's brain. The left image shows their labeled ganglia. Today, I'll be focusing on the cerebropleural ganglion. Their brain has about 4,000 neurons, many of which are large and identifiable. Different brain cell clusters express different neurotransmitters, peptides, and hormones. The image on the right shows vasopressin expression in cyan as a proxy for conapressin. So since conapressin is so well conserved across species, and reproduction is vital to an animal's fitness, are vasopressin reactive brain cells an indicator of size and condition in Berghia? We found that brain size correlates with body size, as you can see by the trend line in the graph. We found this by measuring the cerebropleural length and body length of the slugs using Fiji software. We also found that larger brains tend to have more AVP reactive cells. AVP is the abbreviation for vasopressin. We counted the number of cells in the medial cluster of the cerebropleural ganglion, shown in the white box, and compared it to the cerebropleural length. Another result we had is that as the brain grows, the size of the AVP reactive cells increases. We measured the AVP expression of the medial cluster through the brain slices, which is the Z-plane, to estimate the total volume of the cluster, then divided by the number of cells to get the volume per cell. We then compared the volume per cell to the cerebropleural length. We then went on to investigate how animal condition may affect vasopressin expression and brain size. We did this by taking a regularly fed communal tank of sea slugs and forming them into two groups. The starved group was in a tank with other slugs but no food, and the isolated group was alone but did receive food. We found that the brains of Berghia decreased in size proportional to the time that they were deprived of food. You can see the negative slope of this graph, which compares the cerebropleural length and the weeks of starvation. By comparing the number of cells in the medial cluster expressing vasopressin to the cerebropleural length, we found that the number of AVP reactive cells does not correlate with brain size in starved slugs. When we looked at the isolated animals, we found that slugs isolated for longer durations have larger brains. We also found that the number of AVP reactive cells does not correlate with brain size in isolated slugs. From these experiments, we can conclude that AVP immunoreactivity can help us explore conapressin expression. AVP expression does not consistently vary with starved or isolated conditions, but brain size does. And since AVP expression correlated with brain and body size, conapressin may relate to growth in Berghia. Thank you for listening to my presentation, and I hope you learned some cool stuff about brains, hormones, and sea slugs. Oops.